So I could not fold a paper crane to save my life. And I know this because I tried last night in preparation for the talk, and it ended up as a crumpled ball in the corner of my living room. But in each of my cells and in each of yours are these expert origami artists capable of folding the most intricate molecular machines. And these are tiny gadgets called proteins that perform almost every essential function in our cells. So, as introduced, I studied proteins, protein structure, and protein dynamics at Wits University in Johannesburg, the wonderful city of Johannesburg. Then I became interested towards the end of my PhD in how proteins help other proteins to fold. And for this, I kind of grew a beard to protect my face from the cold and moved north to Germany, around Munich, to the Max Planck Institute of Biochemistry. And in 2020, I moved here to start my lab at this fantastic institute to study how proteins begin to fold as they are made. And these concepts will, I think, become clear over the course of the talk. So first of all, what are proteins? Now, many of you probably think about proteins as the things we eat to keep ourselves healthy as part of our diet. And this is true. But one of the things that the proteins we eat is good for is to make these little molecular machines. And this is how a biochemist thinks of proteins. So proteins are these intricate folded structures that perform specific functions in our cells. And they really are quite remarkable in the shapes that they adopt. So here are a few examples of ones that I just thought looked particularly interesting. And these are experimental structures, so that scientists have determined what these proteins look like in their state in cells. And importantly, this is all on the nanoscale. So these are sort of nanometer structures, 1/1000th of a meter in size. The one on the left is involved in transporting cholesterol in our blood. The next one is called actin. It has to do with keeping our cells in their correct shape and allowing our cells to move. Next, we have something called the apoptosome, this fantastic sort of flower-shaped structure. And this is involved in programmed cell death. So when our cell sort of gives up, then it dies. And on the right-hand side is a protein called ATP synthase that makes the energy we need for our cells to do work. And this is also a fantastically complicated structure with a rotor spinning on a kind of on a stalk. So proteins are critical for essentially every process in our cells. The aficionados in the audience may not recognize this as the outline of a cell, this fried egg thing. But proteins are involved in maintaining cell shape, in performing cell metabolism. For example, all enzymes are proteins. They're also involved in replicating our DNA, our genetic material. And they're involved in producing the energy we need to do all of this in mitochondria. And what's important is that protein fold is absolutely critical for protein function. So proteins only work when they have the correct shape. And this shape then determines how they work. So for example, over here, this is the cytoskeleton, so it's the cell skeleton, part of the structure that tries to keep our cells in the correct shape. And the protein we use for this, or one of them, is called actin. So actin is composed of several parts, and they fit together into these specific arrangements that allow them to grow into long filaments. And the shape of these filaments and the size of these filaments helps to determine our cell shape and allow our cells to move, and also allow things to be moved around our cells. This is the protein I showed on the previous slide, this ATP synthetase, which sits in the mitochondria and helps to produce energy that we use to do other cellular processes. And this is also a fantastic example of how fold determines function, because the top part of this actually spins like a rotor on the rod, and it sits in the membrane and makes ATP. So these structures come from somewhere, and they all start off as long linear chains of amino acids. So proteins start their life as this kind of floppy cooked spaghetti, right? And this is made of sequences of amino acids, so little molecules which are strung together in a particular sequence to form this chain, which ultimately is the protein molecule. The order of these amino acids and the type of amino acid then determines what the protein will become, how it functions, and how it folds. And you can sort of imagine for a simple cartoon protein like this, how one might get from this, floppy spaghetti to this folded thing. You can form these spirals, you can start to pack sheets together. It's much more difficult to imagine when you come to a real protein like this, ATP synthetase, which is gigantic, composed of many parts, and has much more intricate folds. So what then is protein folding? Now, the field of protein folding was really catapulted back in the 1950s by one simple and kind of very elegant experiment and one very insightful thought experiment. The experiment is from Christian Anfinsen in the 1950s. And what he did was to take a fully folded protein out of cells, add a chemical called urea, which unfolds it completely, so it removes the structure, it stretches it out. Then to take this and remove the urea, And what he noticed was that it was able to recover its original state. And this remarkable experiment shows us that all the information needed to get from the unfolded state to the folded state is in principle encoded in the protein already. It doesn't need any external factor to help to reach that state. The thought experiment was in thinking about how this might actually happen. So what Cyrus Leventhal thought about was how a protein might go from the unfolded state to the folded state. And one option, sort of like the monkeys and the typewriters, is to just try every possible combination until you hope you get there. So what he calculated was that if the protein simply sampled every possible state, very fast actually, it would take longer to reach the native state than the age of the universe. And in fact, proteins fold normally within a second or so, which means that they do not fold by just randomly trying out every combination, but they fold by following probably specific pathways. And we often think about pathways using diagrams like this. So this is what we would call sort of an energy landscape or an energy diagram. But the important point to remember is that all the little wells in this are stable states along the way. So instead of trying to get to the folded state all at once at the bottom of the landscape, instead you can make these partially folded states, and one then leads to the other, leads to another, and eventually you get to the native state, or the folded state of the protein. And this, we think, is mostly how proteins fold. So they don't sample every state, they don't try to collapse all at once, but they gradually go through specific partial folds, like folding one bit of the crane, then the next part, and the next part. But the problem, maybe as you can already appreciate, is it's quite easy for this to go wrong. So say you put the wrong part next to the right part, and you gradually end up with something that is slightly different from the correct intermediate. You then end up with something which is misfolded, so incorrectly folded, or even clumped together, forming what we call aggregates or clumps of proteins. Even for a cartoon model, this is rather a complicated process. But for a real model like this apoptosome over here, it's very difficult to imagine how you could navigate this landscape. Which intermediates do you go through? How do you avoid getting trapped in these slightly incorrectly folded states? So what then happens when folding goes wrong? So what we know is that the proteins sometimes fail to fold correctly. You start at the unfolded state, maybe you get to the correct state, but you can also get to something which is slightly incorrect, and this is enough for it to stop working. If you remember at the beginning, I told you that fold determines function, and it has to be exactly correctly folded for it to be functional. But even worse, you could have something where instead of folding slightly incorrectly, these unfolded bits of protein can clump together. And this is very easy to imagine. It's happened to all of us, I guess, maybe no longer. But if you have corded earphones and you put them in your pocket, as soon as you take them out, somehow, inevitably, they clump together, they are knotted, they are tangled. And this is exactly what happens to proteins if they spend too long in this unfolded state. And these are particularly dangerous, especially in terms of disease. So these protein tangles, these aggregates, are associated with Alzheimer's disease, with Parkinson's disease, with Huntington's disease, and many other pathologies. And there are many other types of protein misfolding that cause disease in people. And it's very easy to understand why. There are three causes at least. The first is that the protein no longer works. So maybe that protein is an enzyme, which is important for us to sort of perform some biochemical reaction in our cells. If the protein is slightly incorrectly folded, it no longer has that job, right? It can't work anymore. The other is that these tangled proteins are themselves toxic. So not only do they no longer work, but they also have a toxic effect on cells. They suck up other bits of the cell, other proteins which you otherwise would need. And they also can sometimes form these ordered structures which puncture the cell membranes and otherwise disrupt the cell. And what this leads to is what we often think of as a vicious cycle in protein aggregation, meaning that out of bad luck, you have a little bit of protein aggregation. That protein aggregation makes the cell damaged. It sucks up things the cell needs to stop more protein aggregation, and as a result, the protein aggregation only increases. So if you get a little bit unlucky, you can end up with a vicious cycle that the cell gets worse and worse and worse. So obviously this is a challenge for our cells to solve. But I think one of the remarkable things is the scope of the problem that they have to deal with. You could imagine that if it was your job just to fold this one protein over here, it looks quite complicated, but you could become an expert at this, given enough time. But our cells don't just have to fold this one protein, they have to fold this huge array of proteins, tens of thousands of them, which all look very different to each other, have different sequences, have different structures to get into. So somehow our our cells have to solve the folding problem, not just for one type of fold, but for many. And they have to do so incredibly efficiently to prevent us becoming getting protein aggregates, protein dysfunction, and leading to disease. So that comes to really the focus of my lab's work here at the Crick, which is how cells fold proteins. How do our cells make sure that proteins fold correctly, considering we really, really want to make sure that they do? So first, I guess we have to take a step back and just remind you, if you don't know, how proteins are made. So this slide here depicts the flow of genetic information, which is actually an idea, I think, that is credited to Francis Crick, so fittingly I should put it on the slide. And this says that the DNA in our cells, our genetic information, is transcribed to form a messenger molecule called RNA, and then RNA is then translated to form the protein. So the protein is a product of our genetic information, and this flows from DNA to RNA to protein. Now, the important part for this talk is this last step, how we go from the message, which is the sort of transcription of our genetic material, to the protein. And this translation step is done by a machine called the ribosome, which is itself a giant collection of proteins and also RNA, one of the more complicated protein molecules in the cell, and it has a very important job, and that job is to read our genetic material and produce proteins. Now, what is amazing, I think, is the sheer scale of protein production in our cells. So these ribosomes, which are responsible for making proteins, this is what they look like. This is now a kind of real data showing roughly what the shape of the ribosome is. Each ribosome makes around one protein every 80 seconds, approximately. And a single human cell contains 5 to 10 million ribosomes. And actually, this can be visualized. So on the right-hand side of the slide, this is an image showing the periphery, so just the edge of a human cell, just a little corner of it. And what Tom in my lab has done is to use one of the fancy electron microscopes, which are in the basement of this building, to take images of the edge of the cell and identify the ribosomes and figure out where they are within this part of our human cell. So what we're doing now is to sort of tilt the image to see different views of the cell. And then what I'm going to show you shortly is where all the ribosomes are within this little bit of a human cell. And I hope what you can appreciate is that our cells are absolutely full of ribosomes. There's actually very little room for anything else. So millions of ribosomes in every cell, and every ribosome is making one protein around every minute and a half, which means that our cells synthesize around 4 to 8 million proteins every single minute, and they do this constantly. So cells, therefore, must be extremely good at folding proteins. We must be experts at it. How then do we do this? And we think one of the most important ways, and something we study in my lab, is the way that proteins begin to fold as they are being made. So this slide shows this ribosome moving along the mRNA, so the message. So remember, the mRNA is the transcript of our genome. It's the code specifying what type of protein is made. So the ribosome has two parts, this large part which sits on top of the mRNA, and the small part just underneath. And as it chugs along the mRNA, it stitches together the correct amino acids to gradually form this protein chain. The chain moves through what we think of as the exit tunnel of the ribosome, and then gradually starts to emerge as it's born into the cell. And then we think it can start to fold. So it can gradually start to sort of acquire more and more folded states, slowly as more and more of the sequence is synthesized, until eventually this protein is free to go and do what it needs to do in the cell. And hopefully this all works correctly. We're interested in studying this process and in understanding exactly what these states are that form as the protein comes off the ribosome. Experimentally, this is quite a challenge. So when you study proteins, normally they're easier if they're small and if they're like rocks, like really stable. And this is neither. So on one hand, the ribosome is a huge system composed of more than 50 different proteins that come together in a specific arrangement, so it's large. And the other problem is that it's flexible, it's dynamic. And it's dynamic in two ways. So one is that the protein that's being made as it comes off the ribosome is dynamic because it hasn't got its whole sequence yet. It can't fold completely. It's just starting to fold. It's still flexible. This is very difficult for us to experimentally measure. The other problem of dynamics is that the ribosome is moving. So it's moving on the mRNA, gradually reading the genetic code and producing the synthesized protein. We can deal with at least one of these problems with a little experimental trick that we use very often in the lab. So what we do is to engineer the mRNA, the code, to add in a sequence that once it's synthesized, stalls the ribosome. So the ribosome chugs along the mRNA. When it reads that sequence, it recognizes something is wrong and it stops. We can then purify this whole complex set of cells that has the ribosome and a partially made protein hanging off it. And we can then do experiments to ask how folded is that protein at this stage in synthesis, what recognizes it in the cell, and what is the fate of this ultimately. And we can do this at many different positions to try to get an idea or to build up an idea of how a protein folds as it's being made in the real context of folding in the cell. What I'm going to do over the next few sections is go through the different ways that protein folding can go wrong specifically, and then how we think the ribosome works to prevent this from happening. So how the ribosome allows proper folding to happen in our cells. So the first is that large proteins can fold slowly. So if you have something rather small, you could imagine that it can very quickly be folded into the correct state. If you have something much larger, the problem is finding that correct fold. And this can take a while if you're trying to fold all at once. And the solution is to, instead of trying to fold all at once, you rather fold in a series of very small steps. Each step is individually easy, and if you add them up together, altogether, this gives you the correct structure. So this now shows some of our experimental data supporting this idea. So this is kind of a schematic view of a protein emerging from the ribosome as it's being made and as it's starting to fold. And the bits that are colored red are the ones which are not yet folded, and the bits that are colored in blue and teal are the ones which are beginning to fold. And this is sort of our model based on our experimental data of how this might be happening. So what we think is that at the very beginning, sort of on the left-hand side, the protein comes out of the ribosome, is not yet folded. Then gradually you start to get little bits of folded structure forming sequentially, bit by bit, as the protein is made. And what's really striking to us is that some of these states that were in the middle, where proteins are partially made and partially folded, these never exist off the ribosome. They're not stable and they fall apart. And we could see this because if we removed this one state off the ribosome, it unfolds. And it turned out the reason was that it has this flexible tail. It's partially folded and then it has a bit of spaghetti at the end. And the spaghetti is enough to sort of destabilize and unfold the protein it's attached to. But the ribosome is special. It has this exit tunnel I talked to you about earlier, this bit in the center which protects the spaghetti during protein synthesis. So on the ribosome, it's protected and you can form this small structure. Off the ribosome, you don't. So the ribosome is a special environment that allows small domains or small parts of the protein to fold as they're being made. And we think this makes the folding process easier in cells. But the ribosome can also help folding directly. So this is a real structure of the ribosome as opposed to the cartoons I showed you earlier. And I'll describe little bits of it so you don't have to be intimidated by this. So the top half is what we call the large part of the ribosome, and the bottom half is the small part. And in the center colored blue is our newly made protein, which now we're visualizing in reality coming out of the ribosome and beginning to fold through the exit tunnel that I talked about just now. And what is amazing is that as this protein starts to come out of the ribosome, it already starts to form these spiral structures, which we call helices, sitting on top of the ribosome surface, meaning the ribosome actually helps folding directly. It doesn't just create an environment for small parts to fold. The surface of our protein synthesis machine is also involved in helping to fold our proteins correctly. So these names, this UL29 and UL23, just refers to two types of ribosomal protein. L is because it's in the large part of the ribosome. They call 29 and 23 because the ribosome has so many proteins it's easier to number them than to name them. And it's U because it's universally present in all domains of life. So this is the same system whether we're talking about bacteria, archaea, or us. And in all of these cases, or at least in this case, we think that this newly made protein can begin to fold using the surface of the ribosome. It helps folding directly. It makes these little spiral structures. So that's for proteins that have single large pieces that can take a while to fold. But there's a different type of problem with proteins that have several parts. So actually, most of our proteins don't have just one piece. They actually have two stitched together, which are separate, are sort of distinctive from each other. And the problem here that we run into is that these two parts, which we call domains, often try to fold at the same time. So it's a problem of coordination, right? So trying to sort of tap your head and rub your stomach at the same time. If you get uncoordinated, these two parts get tangled together while they're trying to fold. So the solution, of course, is to keep these parts separate while they're folding. And we think the ribosome does exactly this. So here's an example of a protein which has two parts, A and B. If it starts off unfolded, maybe those two parts fold properly and come together, or maybe those two parts lose their coordination and become tangled with each other. On the ribosome, this is solved in two ways. The first is just making sure that the proteins come out, or the parts come out one after each other. So this is because fundamental to the way that proteins are made is that they're made in a directional way. One part starts being synthesized and comes out before the other is available. So naturally, as proteins are made on the ribosome, you can fold part A before part B is even there. So there's no chance of part A and B getting stuck together and tangling. But more than that, we actually found that even when A and B had both been made, the ribosome kept these from interacting too stably. It keeps them apart until the very end, later on when they can fold correctly. So the ribosome stops this problem of protein tangles by preventing proteins from interacting too much while they're still being made. Now, another very common type of problem comes when you have proteins made of several pieces. So not two parts of the same molecule, but two molecules entirely. And most proteins in our cells are actually like this. They consist of multiple parts, and we have to stitch them together into the correct arrangement. They could, for example, form this one structure that you can see on the right-hand side of the screen, or maybe if you get it wrong, you can form a different type of structure. And you'll all probably be familiar with this from trying to put together a piece of furniture from IKEA. When the parts often look essentially identical, you put them together, you think you've done it right, but it turns out it's the wrong way around. You have to take it all apart and start again. The answer, of course, is to follow the instruction manual. But what the instruction manual will do to actually solve this problem is to tell you what order to put the parts in, which one to start with. And if you start in the correct order, you probably won't end up with two parts that fit in multiple ways. So what we think the ribosome does is to create a unique order of assembly, which means you can only get one type of structure at the end. And actually, what we and many others have found is that ribosomes or proteins can begin to assemble while they're being made on the ribosome. So if the protein has multiple parts, it doesn't wait until later to come together. This association actually begins at the very earliest stages while the protein is being made. So this shows some experimental data where we tried to look at a protein coming together in different parts off the ribosome. So off the ribosome, what we would like to get is the structure on the left with just 4 pieces. But actually, we get that and these amazing sort of star-like structures, which are completely incorrect, where multiple sections of 4 pieces come together to form a ring. In our normal cells, this never happens, which means the ribosome stops these alternative structures, these alternative assemblies from occurring. And actually, we think this is because assembly is directed during protein synthesis on the ribosome. And this is because one piece starts to interact with another piece while that one is only partially made. And this gives the whole assembly process direction. It gives it instruction. And if you start this way, you only end up with one product, the correct one, instead of these alternative arrangements, which are misassemblies and are not functional. So the ribosome is an excellent start to a protein's life, right? So I've showed you how it can promote the correct folding of proteins. It can stop proteins from going down these incorrect paths and maybe forming tangles or misassemblies or folding slowly. But the ribosome is not enough. And perhaps, just like us, protein folding is chaperoned. And exactly for the same reasons, right? To prevent, say, inappropriate interactions. So our cells are full of what we call molecular chaperones, which are proteins that help other proteins to fold. They stop inappropriate interactions, and they make the protein folding process more efficient. And moreover, these chaperones actually act during the process of protein synthesis. So they don't wait until afterwards, they act at the ribosome. While the protein is being made, this is when the chaperones come in. This is when the protein, to be fair, is most vulnerable, right? It's still young, it's not fully mature. This is exactly when the chaperones are required. The difficulty is, especially in human cells, is that there are hundreds of these, about 300 in a typical human cell. many of these seem to do very similar things. They're backup layers. They are sort of levels of redundancy. If one thing fails, you have a backup, and you have a backup for the backup. The other problem is that many of these are essential, so we can't really deal with them. So to deal with this and to understand the basic logic of chaperone-mediated protein folding, or the ways that chaperones help proteins fold, we need a simpler system. And as it turns out, although we think we are special, the chaperone system, and indeed the protein folding system, is almost completely conserved from us to bacteria. Because the problem is essentially the same. Bacteria also have to make proteins. They also have to fold them. Their proteins are no less complicated than our proteins. And actually, one of the most remarkable things about the system, I think, is how well conserved it is. So evolutionists come up with the same idea, or at least it's an ancient idea, an ancient solution to the problem of protein folding, which is to use the ribosome and to use a certain set of molecular chaperones to help proteins fold. So we've taken advantage of this to take a simpler system which lets us understand the basic principles by which protein folding is promoted in our cells. And I'm going to introduce you over the next few slides to the types of chaperones that exist in bacteria, and these are really conserved, most of them also in us, and then how they work. So this first one is called trigger factor. Don't worry about the names. I'm happy to answer questions later on the names. So we often like to, of course, anthropomorphize things. And so we imagine trigger factor has arms, it has a neck, and it has a head. You can't see the arms that well, so I'm going to rotate the structure. You can sort of see these arms like this. People also often refer to it as sort of a dragon-type structure, where it's got a tail, the arms, and the neck. We also have, or bacteria also have, a chaperone called DNA-K, which has two states. This open state, like this, and then closed state, like this. And this is driven by ATP, so it uses energy to open and close. You can think of it as a sort of pair of molecular tweezers. It tweezers over and it closes down on another part of a different protein. And then we have something called the Groeal system. And this also is present in our cells too, in a different form, but it's a similar type of structure. And these are quite amazing because they form these essentially barrel-type structures. So from the side, you can sort of see this barrel shape. And if you look from the top, you can see the hole in the middle. And using another chaperone called GROES, you can cover the hole. And this cavity that's formed inside is actually big enough to engulf an entire different protein. So this one acts as a chaperone by completely encapsulating a different type of protein. So we have three here quite different types of chaperone that perform the job of protein folding in bacteria, and very similar types in our own cells. We have trigger factor, which sort of has the arms for like hugging the protein. We have DNA-K, which has the tweezers for clamping down on small parts of it. And we have GROEL, which simply engulfs the whole protein and helps it fold in the center. And we've learned quite a bit about how these work using these bacterial chaperones. So we know that what trigger factor does is it sits directly at the ribosome, waiting for the new protein to come out and be made. And what it does is to bind those parts of the protein that are not properly folded and otherwise sort of keep the ones that are folded happy. We also have DNA-K, which comes like a pair of tweezers and latches on to the parts that are just not completely folded yet. Latches on, lets go, latches on, lets go, and gradually the protein begins to fold while the DNA-K is sort of binding to it. And then we have the GROEL system, which recognizes these proteins when they are sort of quite unfolded, engulfs them completely, and allows them to fold inside the special cage. So we have this kind of amazing set of proteins whose job it is to help other proteins to fold correctly. And actually, if you put all of this together, we can take a few lessons from the experts in parenting. And they are the experts because they raise 1 million new proteins every minute. So when the protein is young and just coming off the ribosome, it doesn't need any chaperones. The ribosome itself is chaperone enough. It's a good home for the young protein. It lets it grow up initially, in a nice state. It's got everything that it needs. As it gets a little bit longer, trigger factor is the first chaperone to come by. And trigger factor really is the ideal parent. It's attentive, but it's not overbearing. It recognizes those parts of the protein that are slow to fold. It delays their folding a little bit more, but it doesn't hold on too tightly. It lets us start to fold into whatever shape it wants to fold into without sort of preventing it from getting there. The other thing trigger factor does is to protect this protein from the other chaperones, DNAK and GROEL. As the protein outgrows trigger factor, this sort of now adolescent protein starts to encounter DNAK. And DNAK is a lot stricter than trigger factor. It recognizes parts of the protein that are even slightly incorrectly folded, finds them with those tweezers, and hangs on. It prevents them then from getting involved in inappropriate interactions, as I mentioned earlier, getting stuck together with other proteins, and it stops it from misfolding completely. Fortunately, this protein was raised in a loving home, right, at the beginning. There aren't too many developmental problems, hopefully, and so DNAK doesn't have a lot to do. Just these little bits that are out of place. But of course, you may have delinquents. And this is where GROEL's system comes in. Now, GROEL is the disciplinarian. What it recognizes is proteins that did not fold correctly with trigger factor or with DNAJ and still got to a later stage in their synthesis. Those proteins that are still quite unfolded. GROEL takes those, binds them, unfolds them even more to get rid of any mistakes, maybe from earlier on. But GROEL is not cruel, right? It gives the proteins a second chance. It encapsulates them in the central cavity, and this gives it two advantages. One is that it's now completely isolated from the rest of the cell, which maybe is exactly what that protein needed. And the other is its change of environment. So within GROEL, it's in a confined space, and the chemical environment inside that cavity is also different to what normally one encounter. And this change of environment is probably exactly what this protein needed to fold correctly. And actually, many proteins in our cells and also in bacteria become addicted to this. So they are sort of chronically delinquent, and they always need these types of chaperones to fold. So the last little bit, I'll just talk about some future directions for this research. So I showed you this fantastic image from Thomas earlier, which gives us an idea of the environment of protein synthesis and protein folding in our cells. Cells are packed full of ribosomes, making proteins all the time. A lot of what I've shown you comes from us taking these things out of the cell and then studying them in detail, which is great, but we would love to know exactly how this happens inside the cell. And I put here, we would like to follow it and understand it, because these are not the same thing. We would love to be able to watch what happens in the cell, but that's like watching a car drive by. You don't know how it works. We want to understand how it happens in the cell. So we need to be able to look at it in the cell, take it out, build it up, and engineer it ourselves to truly understand this. And this, I guess, is an ultimate goal. The other thing we'd love to be able to do is to predict the folding process. So many of you probably know about sort of the revolution in protein structure prediction with AlphaFold. So if we know what a protein sequence is, we can, with a lot of accuracy, predict what the final structure would be. But that tells us nothing about how it gets there. And the problem here is we know so little about this process, we're definitely not in position to start to predict it. But ultimately, as we understand more, we would love to be able to predict this process, even for sequences that are new and have never been studied before. Finally, I'd like to thank my wonderful lab, who really are the ones who did all this work, our collaborators at the Crick and elsewhere, and the technology platforms which have enabled a lot of the work that I've shown you here today, our funders, and all of you for listening. Thank you.